The next topic is structure of proteins. Proteins, we know that it is very, very important. For example, hemoglobin, which is responsible for the carrying of oxygen in our body, is a protein. And then you have insulin, the hormone which is responsible for regulation or which regulates the blood sugar level, is also a protein. And then they are also responsible for the building up of muscles and tissues. So from this you get an idea that protein plays a very important role. Now, have you ever wondered how does the structure of proteins looks like? So in this topic we are going to look at the different stages of structures of protein. So for that, first we know that the proteins are made up of alpha amino acids. That is, it is the polymers of alpha amino acids, which means when many alpha amino acids join together, it leads to the formation of proteins. Now for that, you need to understand that the amino acids come together only when a link is formed between the two, and that link or the bond between the two amino acids is called as peptide bond. Like we had glycosidic linkage in carbohydrates, here we are going to study about peptide bond. So let me explain what is peptide bond first. Now for that, I'm going to take the example of formation of peptide bond in glycine and alanine. Glycine has the formula in which R is H. So H, and then you have your acid group, COOH, and then you have the amine group, NH2. This is glycine where R is equals to H, and alanine is when you have R as CH3, and then your COOH, and then the H, and then the NH2. Okay, I'm just writing this NH. NH, H, and its lone pair of electrons. Okay, now these two amino acids are linked through a bond called as peptide linkage. Now how does that happen is, the OH of this amino acid will react with the H of the NH2 of the next amino acid. This OH and this hydrogen will leave in the form of water molecule, that is dehydration reaction takes place. So when you remove a water molecule from this, a link is formed between this carbon and this nitrogen atom. So you will get a product where you have CH, just write this smaller. Okay. You are going to remove a water molecule, and what happens is CHH and then this NH2, and you have C double bond O. Now this carbon is linked to this nitrogen atom, so N and this hydrogen atom, I'll write it on the top, and then you have CH3 H C double bond O OH. Now you can see that an amide linkage is being formed. NH located next to CO is amide. So this amide linkage is also called as peptide bond or peptide linkage. So in this case, a peptide bond is formed between two amino acids. Therefore, this is called as dipeptide, containing two amino acids. Now, if three amino acids are joined together, then it is called as tripeptide. If four amino acids are joined together, and it is called as tetrapeptides. If 5 pentapeptide, hexapeptide goes on, now 10 or more amino acids are joined together, then it is called as polypeptides. More than 10 amino acids. It's called as polypeptides. Now that we know what is polypeptide, protein is a polypeptide because it contains more than 100 amino acids. As it contains more than 100 amino acids, and it has mass higher than it has mass higher than 10,000 mu. So only when it contains more than 100 amino acids and has mass higher than 10,000 mu, it is called as proteins. Now one exception is there in this case. Insulin, which is a protein, this is the exception case. Insulin, which is a protein, is made up of only 51 amino acid residues. So you need to remember this exception again. Insulin is a protein which contains only 51 amino acid, and under the properties of amino acid, there was again one exception where the glycine is the only amino acid which is optically inactive. So these two cases you are supposed to remember. Now this is about the brief introduction to the formation of peptide linkage in protein. And proteins are made up of many peptide bonds which joins all these amino acids together. Okay, now let us look at the structure of proteins, which can be studied at 4 stages. They are the primary structure, the first one. And then you have the secondary structure. And then you have the tertiary structure. And the final one is quaternary structure. So in the case of primary structure, you will see a linear chain of amino acids. So let's say this one circle is one amino acids. Now many amino acids are joined in a linear sequence in the case of primary structure. Here amino acids are joined in a linear sequence. Okay. Now what happens is when this primary structure starts folding, you will get the secondary structure, and this secondary structure on further folding you will get the tertiary structure. Now the backbone of the primary structure starts folding through the formation of hydrogen bonds, which results in the formation of secondary structure. So in the case of secondary structure, there are 2 types. The first one is the alpha helix, or it can have the beta plated sheets. In the case of alpha helix, it is in the form of helical. The folding is in such a way that it takes the shape of a helical. Now all these are amino acids. So let me quickly fill it. These are all amino acids which are joined together by the peptide bonds. So this is a polypeptide chain. Okay. Now in the case of beta-pleated sheets, it is in the form of sheets. So again, these are all amino acids. Okay. Folding of primary structure will give rise to either alpha helix structure or beta-pleated sheets. Now the folding takes place due to formation of hydrogen bonds. Now these two are formed due to hydrogen bonds. These are stabilized by the formation of hydrogen bonds. Now secondary structure, on further folding you will get the tertiary structure. In the tertiary structure the folding is complete. So let's say a complete folding of the polypeptide chain takes place. Now again, these are all amino acids. Okay, now under tertiary structure, again there are of two types, that is fibrous proteins and globular proteins. Okay, complete folding takes place here. And it is of two types. The first one is the fibrous proteins, and then you have the globular proteins. Now the interactions involved in tertiary structure is hydrogen bonds, then there is electrostatic forces of attraction, and also van der Waals forces are involved. So these are some of the interactions which are involved in the formation of tertiary structure. Now in the case of quaternary structure, let's say I have 4 units of this tertiary structure joining together to form this quaternary protein. Okay. Now each of this structure, tertiary structure is called as subunits. If 4 subunits are involved in the formation of quaternary structure, 4 subunits, then it is called as tetramer. If 5 subunits are involved, then it is called as pentamer. If you have 6 subunits, then it is called as hexamer. Example for 4 subunits protein is hemoglobin. An example for 6 subunits is insulin. So this is the overall representation of the different stages of the structure of protein. The simplest form is the primary structure, in which you have a single chain of amino acids joined in a linear sequence. When the folding of the primary structure takes place, it results in the formation of secondary structure. And secondary structure can be of 2 types, alpha helix and beta-pleated sheets. And in this, hydrogen bonds are involved in its stabilization. The secondary structure, on further folding, you will get the tertiary structure, where complete folding takes place. And tertiary structure can be of 2 types, fibrous proteins and the globular proteins, which I will explain now. Now, when many tertiary structures join together, it results in the formation of quaternary structure. Now, quaternary structures can be made of 4 subunits, 5 subunits, 6 subunits. Hemoglobin is an example in which 4 subunits are present. So, hemoglobin is a tetramer, And insulin is one where 6 subunits are joined together, so it is a hexamer. Now let me explain the difference between fibrous proteins and globular proteins. Tertiary structure are of two types, that is fibrous proteins and globular proteins. Fibrous proteins are straight parallel, whereas globular proteins are spherical. And the example for fibrous protein is keratin, which is found in hairs and nails. And also myosin is an example for this, which is present in our muscles. Now, example for spherical or the globular protein is the hemoglobin, insulin, which is responsible for the which regulates the blood sugar level, and then you have albumin, which is present in egg, albumin proteins. Okay. Now these are some of the examples of globular proteins and fibrous proteins, and the difference is it has a straight parallel structure, whereas in this case it is spherical. And one more difference is the fibrous proteins are insoluble in water, whereas the globular proteins are soluble in water. So you need to remember the difference between fibrous proteins and globular proteins. Also, you need to remember the interactions involved in secondary and the tertiary structure. These are important. So, dina harila olana protein ki structure di ngazo ewa shira che thango di primary structure che, dine secondary structure che, dine tertiary, dine quaternary structure lani. The primary di khade lana ko the simplest form today. Sequence chiki nalo la ani amino acids sama tu shaigo de la primary structure che. The primary structure the fold chigara china. Folding the khade re hydrogen bond form chani ko the fold chagi ba. Fold chagi sabala on the secondary structure re. Secondary structure nalo la in a new alpha helix structure ta beta plated sheets ade. Alpha helix adi sabala helical form nala che. Beta plated sheets adi sama kora sheets ki form nalo la ma leble chini res. The alpha helix, the beta plated sheets ni ya stable chingi di hydrogen bond interaction che. Dene also secondary di yang fold chigara china also the tertiary structure ba. Tertiary structure ki nalo la ya ena ni fibrous proteins ta globular proteins ani. The tertiary nalo la interactions khari ni chungo na hydrogen bonds, electrostatic forces of attraction, and then water walls forces of attraction dizo che. The novel ever knew about the fibrous check the globular proteins that the fibrous that the straight parallel chain nervous and globular protein that the spherical nervous. The Korani Kebar Kaduna fibrous the water soluble, water insoluble check, globular the water soluble. Fibrous kinano example the Kaduna keratin landing also Tadam, Simudikina keratin and the myosin that muscle now. The name also globular proteins kinano la hemoglobin, insulin. And the albumin, this is globular proteins. The tertiary proteins, mango chingam do divarachina, quaternary structure of protein. Quaternary structure the tertiary structure chikla subunit chikleba. Subunit she chigore, two chigore, not chigore, she chigola tetramer che, not chigola pentamer, and two chigola hexamer. And tetramer gajinam, you common example chikarana, hemoglobin che. Hexamer is a common example insulin. So with this we are done with the structure of proteins. Now let us know about what is denaturation of proteins. Now you need to know the meaning of native proteins. Native proteins are the proteins which are found in our biological system in its proper shape and function is called as native protein. So it is defined as the proteins found in a biological system with its unique three-dimensional structure and biological activity is called as native protein. A protein can lose its structure, can lose its biological activity when it is subjected to some change. So that process is called as denaturation of proteins. Now, denaturation of proteins takes place when it's subjected to some change like physical change, like change in temperature, or when it is subjected to chemical changes like change in pH, or when it is subjected to strong acids or strong base. During this process, the quaternary structure starts disrupted and results in the formation of the tertiary structure. The interaction in the tertiary structure is also disturbed, which gets converted to secondary structure. The secondary structure also gets disturbed too and results in the formation of primary structure. Tertiary gets disturbed to form quaternary, gets disturbed to form tertiary. Tertiary forms secondary, and it finally results in the formation of the primary takes place. So here, unfolding of protein happens, because of which it loses its activity. This is called as denaturation of proteins. The common example for denaturation of protein is boiling of egg or coagulation of egg on boiling. The coagulation of egg on boiling is a result of denaturation of the egg albumin, which is a protein. But when denaturation of proteins in our body takes place, we suffer from different kinds of diseases. So this is about the native protein and denaturation of proteins. Now the final topic under protein is enzymes, and from this it is clear that enzymes are made up of proteins. They are all globular proteins. And the role of enzymes in our body is it increases the speed of reactions that happens in our body. So they are also called as biocatalysts, which coordinates the different types of chemical reactions in our body. Now, how does enzyme helps in increasing the speed of the reaction is by reducing the activation energy required for the reactants to get converted to products. So let me explain that with the help of this graph. Let's say this is x-axis and then you have y-axis. Here energy required, here the reaction progress. Now, in the absence of enzyme, the reactants required, let's say, this much of activation energy to get converted to products. See, this is my reactants, and only when it has certain amount of energy, this is the activation energy required. Only when the reactants crosses this energy barrier, it gets converted to products. So in the absence of enzyme, let's say this much of activation energy is required. This is when there is no enzyme. now let's say I'm going to carry out this reaction in the presence of enzyme. Now during that, it reduces the activation energy required to such a level that the speed of the reaction is much faster. So this is in presence of enzyme. So that's how the rate of the reaction is increased by reducing the activation energy required for the reactants to get converted to products. It reduces the activation energy required. And there are different types of enzymes present in our body. For example, maltase, which helps in hydrolysis of maltose to glucose, and there is sucrase, which helps in breakdown of the sucrose to give glucose and fructose. So this is about enzymes, and also we have come to the conclusion of proteins. Proteins ki nalo la jangyo dige do ji shinar ba thamonya la proteins dige sama alpha amino acids ki sugyo ema che ani amino acid lana khande chila la hor lana NH2 ta COH ni ja ego ina amino acid le ba ani alpha amino acid la di sambala kisi NH2 the alpha carbon atom kala yo rachna alpha amino acid che. The alpha carbon atom di khadire la na so COH ki nalo la C ki so la NH jo ba di la alpha carbon atom che di ga NH2 yo ba china alpha amino acid. The amino acid ki nalo la R group di jo ke china ani ga so di pa we amino acid ma pu chira ba ga so thare che di glutamic acid che glycine alanine lysine du so shi chire dene ga so classification of amino acid. Amino acid they classify chitani or thambo the basic the acidic character latani acidic amino acid, basic amino acid and neutral amino acid lani. Like kisi NH2 ta COH ni ki thaka de chikwa ina neutral che. NH2 ki thaka de manga ina basic che. And kisi COH ki thaka de manga yoga china and acidic level ba. Then classification chitani wa the essential the non-essential amino acid. Essential amino acid then so khadire na so subuna Masuni joba, Sugi Midu Sambalani, as a chilo nilora, Zola essential amino acid. The non essential amino acid la Sambala, as a supona Sugi du Sambalani, chilo nilao marva, Zola, non essential amino acids. The loaded nalola amino acid nisho nalo ne star mom, Chido Sama Karina essential amino acid. The name also correct the properties of amino acids. Properties nalola tambodi, they are water soluble. Yuvadi colorless, Subadi Karirana, they have high melting points, Shivadi, they are crystalline in nature, and if they get courage in Relna, Zuterine form Chigu Disamala, Zuterine Lana Katechina, Disamala, molecule Chikinala, positive ion, the negative ion, Nicha Ecochila, Zuterine layers. In the overall, you charge the Karirana, neutral. The amino acid Nalola, NH2, the base, COH, the acid bar, acid reduce Samala, quick acid K, the H plus take it one, and the H plus Chego dia base ki group jo ba NH2 digi khare chila NH plus dia le ne ko NH3 plus tan ngo CO minus chagyo di la khale na deuterione and deuterione ki kejo khare na amphoteric in behavior ko acid ta base ni je yam do reaction resani kisi acid nyam do reaction na khare form chare na ngo la cation chira res base nyam do reaction na anion chira resani to che then property of the optical activity nalola Tama amino acid chazanchi optical activity tengu bacha ne madingi di chinanchu resulta na glycine la glycine nalo la chiral carbon atom le washa ba sama nalo la chiral carbon atom res thus dening also correct na structure of proteins ani structure of proteins nalo la peptide bond the condense form chagi ne amino acid ni kima la bond chagi ba condense chagi na COH ka ji OH ta ani Hajo amino acid digi H NH2 nalola H ba korangi dune water molecule chima ne dini kimala bond che ba dila hare peptide bond che ta protein sama kare na polypeptide amino acid gale ma kyo che ani ko gi mass ni kare 10,000 le ma mu le ma ba china and these are proteins like this dini ko gi structure la eva shi ba ta the primary structure eva the secondary suba the tertiary structure shiva the quaternary structure. Primary structure nalola amino acid sama linear sequence ka yo check that is simple sachet ho juba ta kisi linear sequence the fold share also secondary protein structure secondary protein structure the kharichin chakolana ko fold chidi sambala hydrogen bond ki through the fold chidi sambala ani secondary structure nalola ena year ba yana ko alpha helix ta yana ko khari form chelana beta sheets ane alpha helix beta sheets ni cha nalola khari na hydrogen bond biche ta the younger secondary the younger fold chinan ngaza tertiary ragres tertiary nalola ya ena year fibrous protein da globular protein ane and tertiary ki nano interactions kare gulana hydrogen bonds van der Waals forces of attraction electrostatic forces of attraction and dige che the fibrous now globular ni la kheba kharelana fibrous dige na la straight parallel che globular the spherical and fibrous is kharelana they are water insoluble keratin the myosin also ta ta simuna la keratin la ko chuna la shure me this water insoluble and globular nano la hemoglobin albumin insulin so chuna shure this they are water soluble che dene also tertiary mango jinyam do duna kare chelana quaternary structure nalo la tertiary unit chikla sub unit chikla ba ta sub unit shi chigyo yore nga chigyo re shi chigyo la tetramer nga chigyo la pentamer drop chigyo la kare hexamer tetramer ga common example kare chelana hemoglobin che hexamer ga kare chelana insulin che those structure of protein is hot dene denaturation of proteins la chire denaturation of protein la dit sambala ngazo native protein chirba native protein na protein kona sopta da function that sama takta chiro biological system na native protein ta ko chemical change the physical change la dit sambala ani ko di folding it sama ya shing ne unfold chide sambala quaternary the tertiary the secondary the primary la good ba didi ani khare chona protein kona ge lega che ne ba de khare mya cha ho sta di de protein chilani denatured protein levels and then also khare enzyme chire enzyme na biocatalyst which catalyzes the different types of reaction na body also supona reaction na boje go de sama reaction go ro na nge chirwa dige la khare le na enzyme biocatalyst and enzyme ki reaction the khandes go ro tang go na activation energy the neurotane activation neurotane de sama ni react in the product la cha le la ba those enzyme ki any reaction is go ro tang go and those protein that's all now let's move to the next topic, that is vitamins. The third biomolecules that we are going to study under this chapter is vitamins. You might be familiar with this term and you might also know that deficiency of this causes some or the other type of disease. Now under this topic, we are going to study about what are vitamins, classification of vitamins and deficiency of diseases of these vitamins. Now to begin with, what are vitamins? The word vitamin is coined from the term vital amines. Before it was thought that all the vitamin contains amine group, but then they realized that most of the vitamins does not contain amine group. Therefore, the name was changed to vitamins. Now, vitamins is defined as the organic compounds which are required in small amounts for a specific biological function. So let me write it down. They are organic compounds required in small amount for a specific biological function. And there are different types of vitamins, and each of these vitamins are designated by an alphabet, and they are vitamin A, vitamin B, vitamin C, vitamin D, vitamin E, and vitamin K. And some of them have subgroups as well. So we are going to study about vitamin B1, B2, B6, and B12 under vitamin B. So these vitamins can further be classified into two types, fat-soluble vitamins and water-soluble vitamins. Vitamins can be classified into fat-soluble vitamins and water-soluble vitamins. From the name itself, it is clear that fat-soluble are the ones which are soluble in fats and oils, and the water soluble are the ones which is soluble in water but insoluble in fats and oil. So out of these vitamins, only vitamin B and C are water soluble. Vitamin B and C are water soluble. The rest all are water insoluble. So you have vitamin A, vitamin D, vitamin E and vitamin K under fat soluble vitamins. They are soluble in fats and oils but insoluble in water, whereas these are soluble in water. Now the problem with water soluble vitamin is that it cannot be stored in our body. Because it cannot be stored in our body, as it is excreted along with urine, so therefore we have to make sure that we regularly consume vitamin B and C rich foods and drinks. So this is about the classification of vitamins. Now let's move to the deficiency diseases of these vitamins. Deficiency diseases of vitamins. Now the first one is vitamin A. The deficiency of vitamin A causes a medical condition called xerophthalmia, and it also causes night blindness. Xerophthalmia is the condition where the tear ducts stop producing tears, and night blindness is a condition in which the person loses his vision at night. Here, tear duct stops producing tears. It is also called as dry eye disease. Now the next one is vitamin B1. It causes beriberi. Deficiency of vitamin B1 causes beriberi, and it has the symptoms of loss of appetite and retarded growth. And the next one is vitamin B2. It causes chelosis. Chelosis is the medical condition where the corners of the mouth and lips start splitting. So splitting of corners of mouth and lips. And then you have vitamin B6. It causes convulsions, which is also called as seizures. There is rapid contraction and relaxation of muscles repeatedly. And the next one is your vitamin B12. Okay, let me write down for this first. Rapid contraction and relaxation of muscles. Okay, then the next one is vitamin B12. The deficiency of vitamin B12 causes anemia. That is, your body is not able to produce the required amount of RBC, that is red blood cells. Now the next one is vitamin C. It causes scurvy. You must be familiar with this, scurvy or gum bleeding. Okay, then you have vitamin D, deficiency of which causes rickets in children and osteomalacia in adults. Now in both this condition, softening of bones takes place, that is, the bones loses its strength. Then you have vitamin E. deficiency of vitamin E causes increased fragility of RBCs. RBCs easily gets disrupted, which also causes anemia. And the last one is vitamin K. It causes increased blood clotting time. That is, a person with deficiency in vitamin K loses more blood because vitamin K is important for the clotting of blood to occur. So this is about the deficiency diseases of these vitamins. You are supposed to remember the classification of vitamins and deficiency diseases of these vitamins. So with this, we are done with proteins and vitamins. Now in the next class, I'm going to start with nucleic acid, which is the last part of this chapter. Thank you.